The Life Sciences DNA podcast is sponsored by Agilicium Labs, a collaborative space where Agilicium works with its clients to co-develop and incubate POCs, products, and solutions. To learn how Agilicium Labs can use the power of its generative AI for life sciences analytics, visit them at labs.agilicium.com. Amar, we've got Venkatesh Mysore on the show today. Who is Venkatesh?
Dr. Venkatesh Mysore is the co-founder and CEO of Aikium. It's a biotechnology company focused on developing therapeutics through protein engineering, synthetic biology, and deep learning. He founded Aikium in October 2022. He spent his career at the intersection of AI and drug development. He has held positions at NVIDIA, working on accelerating drug discovery using machine learning, at D. Shaw, and at the AI-driven biotech Atomwise. He holds a bachelor's degree in computer science from the Indian Institute of Technology, a master's degree in computer science from the University of Wisconsin-Madison, and a PhD in computer science from NYU.
And what is Aikium? What is it seeking to do?
Aikium is a startup that is using its proprietary AI platform to develop a new class of drugs to target GPCR proteins. These are targets of great interest to drug developers.
And what are you hoping to hear from Venkatesh today?
I would like to understand how the company's platform technology is able to tackle the specific challenges to find drugs against GPCRs. I would also like to understand the novel proteins the company is pursuing as therapeutic candidates. And I would like to talk about Aikium's business model as well.
Well, before we begin, I want to remind our audience that if they would like to keep up on the latest episodes of the Life Sciences DNA podcast, they should hit the subscribe button. If you're enjoying the content, please be sure to hit the like button and share your thoughts with us in the comments section. With that, let's welcome Venkatesh to the show.
Venkatesh, thanks for joining us today. We're going to talk about protein engineering, AI, and the potential to apply this technology to a target class of proteins called GPCRs. But before we get into that, I wanted to just talk about the announcement today about the Nobel Prize in AI. Can you talk a bit more about that?
Absolutely, Amar. It's a pleasure to be on your show. Yes, today is a big day for deep learning in protein engineering, because the pioneers of the field have been recognized with the Nobel Prize, specifically Professor David Baker. And then from DeepMind, we have Demis Hassabis and my one-time colleague, John Jumper, who we overlapped briefly at the Shaw Research. So it's a revolution in that AI for biology has often been overstated but under-delivered. But this is an example where the impact has been so much that within 5 years of developing the technology, essentially every researcher in the world in the space of proteins is using it on a daily basis. So it's a truly well-deserved Nobel Prize.
Absolutely. That's just really fantastic news. So let's get to the protein engineering that you're doing and the proteins, right? So let's start with like some basics here. So what is really meant by a target? And can you please explain what's a target in the language of drug developers?
Sure. So if you think about what causes disease, so many different things can be wrong in our body. But ultimately, if you pinpoint it down to a cell, within a cell, you often have certain proteins on the cell. And very often in a disease, those tiny proteins sitting inside cells could be the drivers of the disease, or they could be why you have the symptoms or why the disease progresses. So a target is a protein that the scientific community studying the disease pinpoints and says, hey, if you could only manipulate or modify or modulate this protein, then we could potentially come up with a treatment or a cure or a way of managing the symptoms. So a target in this context usually refers to a single protein that has been implicated in the disease. And we are most of the time trying to inhibit the target protein from doing its job. But in some occasions, we are trying to enable it to do its job better.
Yeah, so we're basically trying to modify the function of the protein, right? Either if we think that If there's too much protein and overdoing that, then we're trying to inhibit that. If there is not much protein, then we're trying to beef up if its function, right? We're using the drugs that we, yeah. And so there are some like these families of protein, right? So there's protein like families of proteins, are the proteins similar to that. So there are some of these families of proteins that are more popular. Can you tell us a bit more about like what are some of these most popular families, protein families?
So if you think about a cell, the way I try to see where proteins are is whether they are outside of a cell, whether they're on the cell surface, or whether they are inside the cell. And when you go inside the cell, you can ask, is it in the cytoplasm or is it inside the nucleus? So the target classes that are of biggest interest to Aikium are the ones that are hiding in the membrane. So these are protein
Mhm.
targets where they are not accessible. They are buried, embedded in the membrane by design. So they are receptors, signals that respond to signals in the extracellular environment and pass on that signal to the intracellular environment. So we have protein families called ion channels, protein families called GPCRs, and those are the ones, broadly speaking, multi-pass membrane proteins that we
Okay.
think the next big frontier and to be attacked or targeted with protein biologics.
Okay. As a multipass, you mean like they just go in and out like that, right? So within
Exactly.
the membrane. Yeah.
So in
Yeah. And yeah.
just being crossing the membrane once, they go in and out of the membrane. So they expose very tiny regions outside the cell. And now we are trying to bind to those tiny pieces of the target protein that are accessible
Mhm.
outside the cell.
Uh-huh. And so these are signal passers, right? So they basically get some signals from outside the cell, and then they pass the signal inside the cell based on what's happening outside, what the cell should do, right?
That's correct. They are key in
Mhm.
cellular signal transduction.
Okay, okay. So tell us about this GPCRs because you have a lot of focus on those. How many proteins are in this GPCR family? How many already have drugs against them? And are there some like well-known drugs against GPCRs that are out in the market?
Absolutely. So the GPCR class, it stands for G protein coupled receptor. It's one of those, wonders of evolution. Let's say it has created this family of machines that sit on the membrane, and they are attached to what are called G proteins on the intracellular side. And in response to various signals, they undergo slight change in their shape, also called as conformation. And that is sensed by the G protein that is attached on the intracellular side, and then those G proteins go as second messengers and go and deliver the signal. So in this category of targets, in this category of proteins, there are more than 600 GPCRs in the human proteome, and
Mm-hmm.
about 300 or 350 of them are considered validated drug targets. And within that, what you would not believe or what is not common knowledge is that we have approved drugs for about 150 of those. So
Right.
GPCRs, 1 in 3 or 1 in 4 drugs are actually targeting some GPCR or the other. The most popular one in 2023, 2024 is the GLPs that everybody is talking about in the context of obesity and weight loss. So there, it is a small drug that goes and binds to a GPCR to achieve its function.
That's fantastic. So it's really a lot of revolutionary drugs, right, coming out of the GPCRs. I mean, that's just a fantastic statistic. So many of the drugs targeting GPCRs. So tell us, like, with so many drugs against GPCRs, what is Aikium trying to do by finding more drugs against the GPCRs?
Great question. So to understand that, we first need to ask what are the different kinds of drugs and when do we need which class of drugs and what is the underserved or unmet need. So broadly speaking, if you want to, let's think about blocking a GPCR for now. Let's say you're targeting a GPCR with the goal of blocking it. You can
Mhm.
either use a small molecule. And when people say a small molecule, it's much smaller than an actual protein. And that's your average pill that you pop in. The second
Mhm.
category of drugs are called protein biologics. And really, the field is dominated by one particular protein biologic family called antibodies. So
Mhm.
antibodies are a naturally occurring protein serving a certain function in our immune system. Whereas about 3-4 decades ago, humanity figured out how to repurpose antibodies into drugs, specifically
Mhm.
make the antibody so that it binds to the target that we are trying to block, and that becomes the basis of a therapeutic application. So,
Mhm.
but now if you ask, if when do we use protein biologics and when do we use small molecules? For many indications, most of us would much rather pop a pill than take an injection or an IV. So protein drugs in general require to be administered intravenously or through the skin. And so you're the most common drug in this case is actually insulin. Insulin is an example of a protein, but it's not going after a GPCR. But
Yeah.
the advantage with antibody-based or broadly speaking protein-based drugs is that they stay in the body for much longer, which means you need far less frequency of dosage. And importantly, there are certain disease classes such as cancer where you need much more powerful drugs. And usually protein biologics have a higher safety profile, have a higher chance of getting cleared in the clinical trial process, and ultimately they can enable certain mechanisms such as ADCC, which small molecules cannot. So ultimately,
Mhm.
protein biologics are preferred for certain chronic conditions.
Mhm.
But yet, given their overall advantages, there are only 3 FDA approved antibody based drugs for the entire family of GPCRs and 0 FDA approved antibody based drugs for ion channels. That's another 600, 700 targets. So this
Uh-huh.
is because raising antibodies to these targets has been hard. So that is the unmet need. And so what IKEM has done is rather than trying to rectify the limitations of antibodies or making small technical improvements, we have come up with a completely new protein family, also inspired by a protein family in nature. And we are
Mhm.
projecting that as the next big uh, industry, meaning it's another vehicle that can become a
Oh,
universal binder and go after uh, these multipass membrane proteins using a different
mhm.
approach.
So these are not antibodies. These are different types of proteins that will bind to the GPCRs and ion channels.
That's correct.
Okay, great, great. And you're calling them seeker proteins, is that right?
That's correct. So we are calling them
Okay,
SeqR, seekers. Uh,
okay,
and the short mnemonic is they are sequence specific binders, or you can think of them as seeking a certain sequence and then binding to it and then enabling
okay,
the modulation of the target.
okay. So can you tell us a bit more about that? I haven't heard much about the seeker proteins, so if you can familiarize us with them, that'd be
Sure.
great.
So SeqR is first of all based on a naturally occurring protein family, but it's not. It hasn't been employed for therapeutic purposes the way we have
Mhm.
conceived it. So we have taken an inspiration from nature and re-engineered it, and this re-engineered protein family is what we are calling seekers.
Mhm.
So
Mhm.
these seekers have a fundamentally different approach to binding to a target. So if you think about a protein, in fact, in light of the Nobel Prize, what the Nobel Prize was about being able to predict the three-dimensional structure of a protein, given the
Mhm.
sequence of the protein. The sequence is the chemical composition. But it turns out only about 50% of the protein surface has a rigid three-dimensional shape. The other half, on average, is a highly flexible region by design, and they are called intrinsically disordered regions, or IDRs for short. These
Mhm.
intrinsically disordered regions do not exist in a single three-dimensional shape. Instead, they are meant to sample several different shapes, and they are highly flexible or floppy. The problem is all the drug discovery and drug development machinery, both on the small molecule front and on the antibody front, has been heavily focused on these structured regions, and the intrinsically disordered regions, which represent about 50% of the real estate on protein surfaces, have
Mhm.
largely been underserved. Our scaffold, the seeker, actually
Mhm.
targets these intrinsically disordered regions. So we are
Uh-huh.
unlocking about 50% of the opportunity, which previously didn't have any reliable, robust technology for attacking.
Okay. And so these seeker proteins that you're talking about, are they like uh, do they like can you specify the sequence that they should bind to, or how does that work?
That's exactly right. So if it's a universal binder in the sense it binds to all disordered regions, such a protein would not be useful as a drug because it would
Hmm.
to not just the target that you care about, but everything else as well. So
Yeah.
instead, in the world of a drug development, selectivity, being able to bind to just the target that you care about, is very, very important. And I just
Hmm.
told you that these disordered regions don't have a shape. So what is it that the seeker protein can recognize and bind to? It can actually recognize the linear sequence of amino acids. So it's like you can specify a zip code or an address on a protein surface, and we can then engineer a seeker that specifically detects and binds to that sequence on the target protein.
Okay, okay. For people who don't know, protein is basically made of amino acids, right? So it's a sequence of amino acids. So what Venkatesh you're saying is that these like unstructured regions that the protein has, where like you have this sequence of amino acids, so the seeker proteins are going to go and bind there, right? And then affect the function of the GPCR.
You're correct. So the common model is think of beads on a chain.
Hmm.
Uh, Most of the time, those beads don't have any shape on their own. But occasionally, you can fold them. Let's say some of them have magnets, some of them have charges. They can fold into a three-dimensional shape. So there will be
Hmm.
portions of this chain that have a structure and portions that don't have a structure, and they're highly flexible. They can wobble around. And
Hmm.
IKM seekers can actually bind to those structureless regions through the special seeker protein.
Mhm. Great, great. So, are you now with these? Are you targeting the GPCRs that have already been targeted, or are you looking at the new GPCR? Like, how are you choosing the targets?
So target selection is actually a much bigger deal than we thought, than we realized when we started IKM. So when you think about targets, we would love to be able to say, the new technology has enabled us to go after GPCRs that have never been targeted before. And so we have some
Hmm.
GPCRs in that category where we know their therapeutic relevance based on the recent academic work, but there
Hmm.
is no clinical trial that has ever been done on those GPCRs in the context of certain diseases.
Hmm.
Flip side of choosing these targets is we already have modality risk, meaning we are doing something new on the drug front. But now, if you
Hmm.
also couple a high risk target that hasn't been validated in the clinic, then you're
Hmm.
compounding the risks. So we
Uh-huh.
have tried to balance it by picking a set of GPCRs, all within the same target class, but
Uh-huh.
ones that have been validated, meaning there is an approved drug in the clinic, others that
Uh-huh.
are in clinical trials but nothing approved yet, and the third category where there has never been any human clinical trials conducted on that target, but the evidence is so strong and compelling. So we have tried to spread our bets that way.
Gotcha. And but then when you're developing something against the GPCRs for which there already are drugs, then the bar is going to be much higher, right? Because then your drug needs to probably beat the traditional drug that's out there. Is that correct?
That's correct. So what we have done is there are several indications. So we have tried to pick GPCRs where they are implicated or connected to more than one disease.
Uh-huh.
So it may be the case that the indication is just the word for a disease that a particular drug has been approved for. So it may be the case that a certain GPCR has been clinically validated for disease A, but there is
Hmm.
strong evidence for disease B. But the
Yeah.
fact that in a clinical trial setting in humans, uh, it has been drugged and it has been deemed safe means that
Hmm.
just by drugging the target, you're not going to cause any toxicity. So you're just changing
Hmm.
the disease context. So
Yeah.
what we have tried to do is hedge there as well. We have tried to pick
Hmm.
GPCRs that are implicated in more than one indication. So in
Okay.
one case, we have a GPCR that's implicated in both oncology and an autoimmune disease. In the other case, we have picked a GPCR that is implicated in oncology and also neuroinflammation.
Okay. Okay. And then, so these GPCRs, what are the therapeutic areas that the GPCRs are commonly targeted against in general?
So that's a good question. But because of the broad relevance of GPCRs, essentially they're present on every cells serving certain functions. They've been targeted, like I said, we have asthma drugs that target GPCR, and they all fall under the broad category of inflammation. Then we have indications like atopic dermatitis. We have indications like, again, all falling under neuroinflammation. Then we have several oncology programs where the GPCRs are targeted for two reasons. Either because they are directly involved in the disease, or simply because certain GPCRs are overexpressed or present a lot on cancer cells than on normal cells. So they are often used as payload delivery targets, meaning let's say GPCR X is present only on cancer cells. And then I can take an antibody that binds to, let's say if I could make an antibody that bound to GPCRX, but alongside I attached a drug molecule that would eventually kill that cancer cell. So this field is called antibody drug conjugates. And this principle is often used, is often the reason why some GPCRs are targeted in the context of cancer.
Okay, gotcha. And you mentioned about the obesity as well, right? So that's another therapeutic area.
Another big application that has revolutionized our thinking of or expanded the treatment options for a variety of indications in the last 2-3 years.
Absolutely. So now let's talk about the machine learning a bit. So Aikium has a large scale machine learning platform to screen and optimize these SeqR proteins. And I believe it's called Yara ML2. So can you tell us more about that?
Sure. So the big realization that happened about 3 or 4 years ago is that there were actually two independent sets of breakthroughs that had happened. One in the field of synthetic biology, and the other in the field of artificial intelligence. And both had to do with reasoning about proteins. So in the world of synthetic biology, rather than test one protein at a time in a certain experiment, you could now make millions, billions, and what Aikium has done is trillions of proteins at the same time in a single pot, and then use that to do experiments. On the artificial intelligence front, two breakthroughs. One is the protein structure prediction that we talked about, acknowledged with the Nobel, but the equally impactful invention was the use of or development of protein language models. So similar, just like we all know ChatGPT, similar models have been trained on protein sequences. So these are called protein language models that reason in the chemical language of proteins and not in an English language description of the protein. So this.
So basically, we're talking. So because we have 20 different amino acids, right, and each is represented by a letter. So your protein sequence is a chain of letters, like you said, like the beads, right? Each is a bead that has this one of the 20 letters. So basically, that's the language that now we're using for the large language models, right? So that to be trained, is that right?
And that's absolutely right. So if we have a 20-character alphabet rather than a 26-character alphabet, and so every protein can be described as a single sentence in that alphabet. So what Aikium has done, or the big realization I was saying was, neither piece on its own can deliver the next big step up. So the next big step up in biology is going to come when these two breakthroughs in orthogonal fields are coupled correctly. So what Aikium has done is we started out realizing that, if you build a really good model and you say, hey, my model has 70 billion parameters, you're going to have that advantage maybe for 3 months, because at that very moment, somebody else is training a larger model, or somebody else is coming up with a different architecture that has something else better about it. So model
in a large language model, you mean?
In a large language model,
okay.
or any other model that's reasoning about proteins or any aspect of it.
Okay.
So the model is not a sustainable mode, whereas is a sustainable mode. So what Aikium invested in is to build a protein interaction platform where we could get or screen 100 to 1,000-fold more proteins than anybody else in the world. So that is called the YOTA display. So if you look at literature, or if you look at protein engineering companies in general, the principle is the following. you have a protein and you're trying to input, and the protein, let's say, is the SeqR protein, and let's say it binds to a piece of the disordered region of the target GPCR.
Mm-hmm.
Now you want to improve it. You want to make a really, really, really strong binder. So the general principle in protein engineering is, if you have a rough starting point that sort of works, then we randomly make changes to it and then see if any of those random changes improve the binding to the target.
Mm-hmm.
The question then becomes, can you really do better than random? And that's where the AI comes in. And the next part of the question is, how many can you test? Can you test 100 changes? Can you test 1,000 changes? How many random changes can I make to my starting point and then test them experimentally?
And you're generating these new sequences using the large language model?
That's correct. So Aikium has
Okay.
its own patented generative AI approach for
Okay.
for designing SeqR proteins given any
Okay.
input sequence. So the input sequence is, let's say, a disordered stretch of a GPCR. So we have a chemical specification, and then we feed the input, and out comes a SeqR protein that has been designed. So then we can make that SeqR protein and test it in the lab.
Interesting. Okay.
So in this approach, the way I just said it, we would take one design at a time, synthesize it one at a time, and test if it works. Instead, what we have done is we are changing the paradigm from testing, top 10 or top 100 to essentially generating a library with physical library with 1 trillion different changes to the starting point. So that number, to put it in perspective, the field has been using a certain wet lab technique called yeast display, where you can make 1 million different changes to a protein and test all of them simultaneously.
Okay.
Then came another break. Yeah.
Sorry, when you say test them, is basically, so in different yeast cells, you have these like all these different proteins, and then you're putting like that GPCR, like that you want them to bind to, and see how good the binding is. Is that what you mean by testing?
That's correct. So let's say you're able to make 1 million different versions of a SeqR, and then you GPCR, let's say, on a magnetic bead, and then all of these million variants are now competing to bind to that target.
Okay.
Only the most potent ones, driven by thermodynamics, will be able to bind to the target.
Okay.
And if you then fish out the target and then do some clever genomics, you can then go back and say, hey, this must have been the protein that stuck to the bead, because I was able to establish its identity using a trick that yeast display allows you to do. So you can pull the identity of the protein with the actual protein.
Okay. That sounds great. Okay. Yeah, please go ahead.
Yeast display, you can do 1 million proteins at the same time, and it's been done for a few decades now. Then came phage display, where they pushed it 1,000-fold larger. So in phage display, you can make 1 billion different variants of a protein, and then all of them will compete simultaneously to bind to your target protein. And then you fish out your target protein and check what is bound to it. You've likely pulled out the strongest binders in that billion protein library. So from there, the next technique that came was called mRNA display. But mRNA display has certain limitations. So it really, you cannot make in mRNA display, you can go to 1 trillion. But the problem is that the method as described in literature, as practiced by others, is applicable not to large proteins, but only to tiny peptides. So in context, going back to GLPs, those drugs are short peptides. And so you can do GLP peptide screening using mRNA display. But
so by GLP, you mean the obesity drugs, right? Like those drugs you're talking about.
Yes. Drugs are themselves short peptides rather than large protein. So peptide is like a tiny piece of a protein. So mRNA display has the library size or diversity advantage, but as described in literature, as practiced by others, it's not applicable to large proteins.
Okay.
Where the Aikium team really spent much of its pre-seed phase building an industrialized version of mRNA display that we are calling YOTA display. And this was accomplished by a team with over 125 patents in the space of spatial transcriptomics, CRISPR, and protein engineering. So this is been the bulk of our effort in the last year and a half. And now we have a highly optimized and a robust protocol that has cut down the number of steps, the wall clock time, the overall efficiency of the process. So now we reliably do trillion protein screens against several different targets.
Okay. And this is pretty much like a wet lab approach you're talking about here, right? Like
This
you have the AI, the large language models on the one side, but this is the wet lab inventions that you're doing on this other side as well.
Absolutely. So Aikium is very fortunate to have founders that span both AI and synthetic biology. And we realized early on that innovation in just one side will not really lead to a quantum leap in our ability to do protein engineering. So what we are doing is going back to the darts on the target analogy. We can now throw a trillion darts on the target, but these darts have not been shaped at random. So AI works ahead of time to shape those darts. So the tips of those darts and the other features have been engineered through a computational exploration to design seekers that are likely to bind to the target of interest. So this combination is what allows us to, find really efficacious seekers. Really.
So basically, what you're doing is with your large language model, you're generating a trillion variants, and then you're testing all of those in the wet lab using this new methodology to see which are the seeker proteins that are best binding to these GPCRs. Is that correct?
That's correct. So to the first approximation, that is correct. The only caveat is, if you really have a generative model and ask it to make a trillion designs, a lot of those may be garbage or redundant. So what we do is we have a way of summarizing the hit list, if you will, from this model. And then we use an intermediate optimizer to make them better. And then we infer the library that collectively spans the ideas that have come out of the computational pipeline, so that we have representations of all the ideas that have come out. But we also do certain permutation combinations of the suggested mutations that may not have been present in the original list. But this way, we are doing a very thorough exploration of the landscape.
Gotcha. Okay. And does that happen in like just one single iteration, or are there like multiple iterations that happen for this?
So computationally, we have multiple iterations, but at the end, multi-day computational pass, we have a library recommendation. And then this library is synthesized. The experimental part also inherently has multiple cycles. So what I previously described was the target you use like a bait, and then you're trying to fish out members of your library that bind to the target. So this is called a selection process. But what we do is we do multiple rounds of selection, meaning we make it harder and harder for those who are stuck to remain stuck. So we really pull out those super potent binders. And we also do what is called negative selection, meaning things that bind to the wrong targets, they are discarded. So the library is depleted of members that bind to the wrong things. So we can get both selectivity and specificity in the same experimental assay. But this is still, one computational pass followed by one experimental pass. But very often we have to do 1 second iteration as well. So we take the hits that we have found, then generate libraries, computationally search for, build a library around the hits that we have found, then subject them to another round of mRNA display. And very often that is sufficient.
Okay, gotcha. And now let's say, when you've done this with the wet lab and you've got some of these potential seeker proteins that could be good drugs. So what have you done to validate these further?
Great question. So when we do these experiments, all that we are doing is binding, meaning does my seeker bind to the intrinsically disordered region of interest? Then this binding is happening in isolation, in vitro. It really is not happening in the context of an actual cell. So the validation that we have done is we have gone to cells. We have gone to cells where the target GPCR is present in its entirety under realistic conditions. And in those conditions, we don't just check for binding, because binding in that environment will be harder than just binding in isolation. But after binding, it has to achieve the functional modulation that we care about. So in the case of some of the GPCRs that we have picked, we want to antagonize them, meaning we want them to want to suppress their activity. So what we have measured and validated so far is that these seekers are actually really, really potent, meaning they are able to bind to the target even under cellular conditions and actually inhibit the ability of that target to perform its function. That's the goal in the context of a therapeutic application.
Okay. And how, like, what is the business model around that? Like, are you seeking to discover promising therapeutic candidates and license them to pharma companies, or you take targets of interest from partners and provide them with candidates, or are you going to pursue your own pipeline? Can you tell us a bit more about it?
So our core, value is going to come through our in-house assets. So that is just the nature of the world we live in today. That platform play, while it sounds exciting, it does not command the value of having in-house assets. So what IKM has chosen to do is to have two in-house programs that we can take to the development candidate nomination stage and then to IND and beyond through our in-house pipeline. But platform we have built is really so efficient and has been worked on so much that it would be a shame if we are not able to apply it to other targets. But really, it is unrealistic for one entity to have the resources to pursue 100 different drug discovery or drug development programs. So what we are simultaneously doing is talking to pharmaceutical companies and enter into partnerships where the company tells us the specific target that they care about. It doesn't have to be a G protein coupled receptor. It can be any protein where they previously employed their in-house technologies. It could be small molecule based or it could be antibody based, but perhaps they did not get the candidate that they were looking for. So in those cases, we should be able to apply our technology, the SeqR based approach and the Yota ML squared platform, to be able to give them potent selective binders to that target, which they can then develop into drugs if they have the other desirable properties that they care about. So far we have a very long pipeline of folks we are talking with, and we have one big pharma partner with whom we've entered the paperwork and negotiation stage. So we can expect an announcement before the end of the year.
Great. And these are early days for Aikium. So you're at the Baker Bioingenuity Hub in Berkeley, and you're raising seed funding. So what have been the discussions like with potential investors? And do you think they are more receptive to you as an AI-based drug developer compared to a more conventional type? Can you tell us more?
Sure. So I think our fate changed in September because that's when we got the really compelling cell-based validation. So previously we were highlighting the platform potential and the fact that if you could extrapolate and go from here, you can see that it can have therapeutic relevance. But now we have functional activity data. And in this world, it has to be really, really potent, what are called nanomolar antagonists. And that's what we against 3 different therapeutically relevant GPCRs. Now it's no longer a science experiment. It's no longer, hey, the couple of dreamers are building something new that may or may not work. It actually works in cells. So now the real challenge is, taking it into mice and then eventually to humans. So there, completely different set of skills are needed, and that's what we are actively recruiting. We are talking to several pharma people, current and former, to bring them on in key roles to take this biologic in its infancy to go all the way to IND filing. That's what we are currently adding to our team. But in the context of fundraising, the AI piece is not going unnoticed. So like I said in the earlier as well, a pure AI play, unfortunately, while it sounds glamorous, with the exception of perhaps AlphaFold, it's not going to have an immediate clinical impact if you say, let somebody else do all the validation for me. So you have to come to the realization that a drug development company has to have AI and has to have protein engineering, in our case, on site. And that's what we have done. And this story is really having a lot of takers. So again, we expect a big announcement before the end of the year. We are in diligence with a lot of venture capital firms, but we'd love to have conversations with more folks who understand this space. Interestingly, for listeners, the field of biotechnology, when it is driven by computational methods, people have flipped biotech and are calling it tech bio. So Aikium is an example of a tech bio startup that is incorporating AI, but realizes the importance of ultra high throughput experimentation to generate that label data that needs the that the hungry AI craves for.
And how do you see? So I wanted to ask you about like the name Aikium. Is that something to do with AI, or like what is the origin of the name?
Really interesting story there. So Aikium in two different Indian languages, Tamil and Sanskrit, means the union or coming together, much like yoga. But in this specific case, we picked the name because three different fields, protein engineering, synthetic biology, and deep learning, have to come together to be able to solve this difficult problem of drugging the intrinsically disordered regions. The 50% of the protein real estate that no one has a solution for. If you really even want to attempt to attack that problem, you really have to unify forces. And Aikium means the union are coming together. But after the name was suggested, we didn't really immediately like it. But then when we realized we could spell it as AIKIUM, so then, we had a way of emphasizing that there's an AI first protein therapeutics company. So we enjoyed that name and we adopted it immediately.
That's interesting. And now, looking at the big picture, I mean, it's a fascinating method you talked about, like in terms of AI, bringing in really new areas, new type of drugs, and also applying the new wet biology methods. So, how do you see AI transforming the pharmaceutical industry over the next decade or so?
So AI has several different applications. Again, I was very fortunate that my career between DSHA Research, Atomwise, and NVIDIA gave me different perspectives on both small molecule drug discovery, protein engineering, and the broader space. So if you think about where AI can be used, companies like Recursion and In Vitro are using them to find the targets that need to be targeted. So initially I presented it as though it was a solved problem. Somebody does the biology and says, hey, target X, the GPCR X, go, find something that binds to it. But really there are the disease etiology is quite complex. And there are often multiple ways, multiple routes to reach the treatment. So what some companies have done is, again, they start with an ultra high throughput experimental platform, but then they employ machine learning to reason, for example, reason over high resolution microscopy images or high resolution, protein-protein interaction data to figure out the network of different proteins, how they are talking to each other, and then find those nodes that need to be, suppressed or activated for the profile to resemble a healthy cell rather than a sick cell. So the first application is AI for target discovery. But now, once the target has been discovered, many companies, such like In Silico, Atomwise, what they have done is they've employed methods to say, hey, small molecule drugs, proven method. Now let's just see if we can find a better drug using AI. So this can involve, cleverly searching through ultra large compound libraries, or it can involve generating new small molecules that would fit into the previously characterized pockets. Or what companies like DISH or research are doing is realizing that proteins are not static structures but highly flexible. Rather than use the one structure in the protein data bank or the one structure predicted by AlphaFold, you can do a molecular dynamics simulation to understand the different shapes that that binding pocket looks like and then plug in compounds there. So this is all under the space of small molecule drug discovery. But the real revolution, in my opinion, is on the protein side of things, because protein language models, protein structure prediction have put us in an era of reasoning that was not possible even 3-4 years ago. So
Okay.
that's a big revolution where I think within the next decade, we will see enzymes, we'll see drugs, we'll see so many things around us, which are all engineered proteins. They may have come from, let's say, synthetic biology platforms, such as those engineered by Professor George Church, or they may come from entirely computational pipelines engineered by pioneers such as Professor David Baker, or it could be some new tool or application that comes from DeepMind or other big players in the space. But that won't be in therapeutics or finding that initial drug. But I think what most people are now really concerned about is, what about the rest of the drug development pipeline? Because things that bind on a computer, maybe some fraction of them will bind even in a test tube. But the moment it goes to a cell, already we are not able to predict whether it will bind under cellular conditions. Then when it goes to a mouse, maybe it will bind to the target, but it could bind to so many other things, and it could cause toxicity related issues that we are still struggling to predict. And then ultimately, when it goes in humans, really, so far we have not been able to predict which drugs are going to fail in phase 1 or phase 2 or phase 3. And those are the most expensive failures, because about 10 years and a few $1 billion have gone in, and then you realize you've produced something that is unsafe for humans or has no efficacy in humans, even though it worked in mice or other large mammals. So that's so what is happening is AI is not, we found the target. I have now new methods of AI driven target discovery. Then I have AI driven drug discovery. Then we have AI driven drug development. Then the real question is, how soon can we get to AI driven toxicity prediction and human clinical trial outcome prediction? And so for that to happen, we need much more data. Like with anything else, we need more lab rats to be sacrificed to be able to generate the data. And the ethics of all of this is definitely being brought into question. So there are some people who are trying to find workarounds, and these involve models called organoids. So even rather than use a whole animal, you can take stem cells or other approaches to create small living organ mimetics, and you can use those to do your drug screening, and that will be much more predictive of subsequent failures. And this is also being enabled by high resolution ways understanding what's happening in a tissue or a cell. That's where fields like spatial transcriptomics, where you look at a slice of a tissue and you can say this cell is trying to do this because these proteins are overexpressed here. This other cell that did not respond to my cancer drug, it looks like it has a completely different proteomic profile. So high resolution ways where they're combining information about which genes are currently being transcribed, what is the cell's morphology, what is the overall, what else can we learn about from the person's genome or blood sample or other components. All of that is feeding. And now we have a richer than we can digest picture of each person. And then we have to tie this to whether that person responded positively or negatively in a clinical trial. And as this data set accumulates, as pharma companies collaborate and share this crucial data, that's when the new kind of reasoning that will benefit all of us will emerge. I think we are maybe 5 to 10 years away from saying we can predict some of those things.
Absolutely. Yeah, there's a long way to go. One more question I have for you is you mentioned about reasoning models. Can you explain, like double click on that? Like, what do you mean by reasoning models?
So when we are trying to reason, traditionally it's about reading 10 papers and then coming up with a new hypothesis. Or maybe protein A works because it talks to protein B. And maybe this is the reasoning that emerged from reading different papers. But the kind of reasoning I'm talking about is multimodal deep learning. So when you think about a patient or a blood sample or a cell or a protein, you're going to have, let's keep it simple and just talk about proteins. On one hand, the chemical composition or the sequence protein. On the other hand, the three-dimensional structure or the conformation of the protein. And then you have lots and lots of annotations about what are the cellular processes that this protein is known to function in. That's the ontology. So if you can, so you have a text description of the protein, you have its chemical composition, and then you have its three-dimensional shape. So these are three different kinds of information. But yet you have to be able to digest all three different kinds of information and then compare two different proteins. Or in the case of, let's say you're engineering a SeqR, to compare two different SeqRs and say which one is going to be better for a particular target. So when I say reasoning, I mean being able to bring in different modes or different kinds of information and then using to either predict the small molecule drugs that are likely to work or engineer the proteins that are likely to be therapeutically relevant.
Okay, gotcha. Dr. Venkatesh Mysore, CEO of Aikium. Venkatesh, thank you very much for your time today.
Thank you so much, Amar, for hosting me. It's really a pleasure talking. And hello and thank you to all the listeners here.
Well, Amar, what did you think?
It was fascinating to really see the company being very active in innovating on both sides, right? On the AI side, developing the large language model, and also on the wet biology side, with what he talks in terms of like synthetic biology, right? So creating these displays of a trillion proteins at the same time. So very interesting business model in terms of like having both of those in the same company in a startup company.
GPCRs are an important set of biological targets. Venkatesh noted there are about 150 drugs that target GPCRs today. What's the significance of Aikium's SeqR proteins and their ability to target what he called disordered regions?
Yeah. So one of the things is that GPCRs is a very well validated target family. I mean, we know that the drugs against GPCRs work very well because they're sending the signals down in the cell. So a couple of ways I can think is one is for the targets that are already there. Perhaps these protein bindings, these SeqR bindings, if they can have better binding than what the traditional drug molecules have, they may be more effective drugs than what we have on the market for those specific targets that are for which there are drugs already. But then also some of these GPCRs are the one for which the drugs haven't been there. I mean, for whatever reason, maybe they weren't the great targets, but also maybe they were not very well druggable in the sense that it was hard to find the right small molecule against them. So for those now become pretty open in terms of like, new targets that we can develop a drug against, and again, this new modality and having a lot of effect in curing the diseases. So I see it in both ways.
It was interesting to hear him characterize his risk management approach to target selection on how the company's balancing novel targets versus those that are well validated and others that may be targets of a clinical candidate. Mm-hmm. What do you think about that? What might it say about the consideration for the application of a technology that can produce a high level of novelty?
So I would say that, I mean, that approach seems very reasonable because the new SeqR proteins, which are the drug candidates that his company is coming up with, those are very novel. So as he mentioned, if you have a novel target and a novel drug, there's a lot of risk that people are taking. On the other hand, if you have a well-validated target, okay, there's no risk there, but then you're developing the drug itself. And I also mentioned to him, right, like if for a well-validated target, if he's coming in with a drug, the drug needs to be much better than what's out in the market for it to be effective, right? So that is an extra challenge that's there. But on the other hand, it helps him validate that the methodology is right, the methodology is correct, and that validation can go a long way in terms of even convincing his pharma partners, right? Saying that these are the drugs that we can develop these drugs against these well-validated targets. We've shown that, so now we can develop those against these unvalidated targets as well. So overall, as he's trying to push these into like drug development, right? Either themselves or through partners. All of that data is going to be helping as evidence that the methodology works.
They're using generative AI to make secret proteins, libraries the size of a trillion proteins. These are weeded down and optimized, but at some point, is there a diminishing return? How much of a challenge of using generative AI is knowing when you're working with either too small a data set or one that's too large? When does enough become enough?
It's hard to answer that. I can tell you that it's trillion may not be enough, because what we're trying to do is we're trying to. It's there's so many mutations that are possible, and right now not much is known about these. So at this point, at the beginning, he probably needs to try out a lot of different combinations to see what sticks. I think once the models and the researchers in his company learn more about what kind of sequences, what kind of mutations are giving better results than the others, then they can guide the models in those directions. And he said that they have started doing that already. But I think that's going to be the thing, and that's why I also asked him about are they having multiple rounds, right? Because a lot of times what happens is that computationally you can develop a lot of stuff, but then once you put that in a wet lab and you get some results, then you can improve the computational model with those results and focus in the areas where you're getting the positive results, right? So in that sense, I think that kind of like a recursive way could be very effective. But it's hard to say right now, like what is enough, because enough is when we can get a good drug, right? That's what it's about.
Well, it was a great conversation, and looking forward to our next one, Amar. Take care.
Thank you, Danny.
Thanks again to our sponsor, Agilicium Labs. Life Sciences DNA is a bi-monthly podcast produced by the Levine Media Group with production support from Fullview Media. Be sure to follow us on your preferred podcast platform. Music for this podcast is provided courtesy of the Jonah Levine Collective. We'd love to hear from you. Pop us a note at danny@levinemediagroup.com. For Life Sciences DNA and Dr. Amar Drawid, I'm Daniel Levine. Thanks for joining us.